Hi, it's Sergei from 100x Bio, and in this video I'm continuing my story about my life choices, why I started to do the PhD in Immunology of Infection. So in the first video I was telling about my school years, about university, and about getting into the grad school. And I went to grad school in Russia into a scientific institute that was belonging to Medical Academy of Sciences, and they were working on developing the targeted drug against tuberculosis. So the concept was to attach isoniazid, which is a molecule, compound drug that can kill mycobacteria, attaching it to biologically inert polysaccharide called dextran, and by conjugating them, isoniazid would be delivered to the cells which pick up dextran. And this was known that bacteria, Mycobacterium tuberculosis, can live or reside within the macrophages and survive there. And dextran also can be picked up by macrophages. This was a concept. It did not look to me as very well thought out, very well validated, but there was some in vivo data where this conjugate was more efficient in kind of treating tuberculosis infection in mice, and actually it was not even real mycobacterial tuberculosis infection. It was infection with BCG, which is the strain of tuberculosis which is like less aggressive, let's say, and which is inducing more of immune response. And also this BCG, mycobacteria BCG, they can induce some liver damage and some level of fibrosis, and somehow dextran isoniazid conjugate was preventing the fibrosis. So there was a concept that maybe this drug has some sort of mechanism of action against fibrosis. Let's study that. And it was very interesting and exciting for me to study the new topics, to study the mechanisms of fibrosis, because fibrosis is a huge medical challenge. Whenever people develop, for example, lung fibrosis, there are no proven way to stop it, and there are drugs which can alleviate the condition, but it's poorly understood how to completely prevent fibrosis of lung or fibrosis of liver, and this is a life-endangering condition, and it's chronic, and it usually tends to get worse instead of getting better, and people can suffer for years. And people who have tuberculosis, they actually can develop lung fibrosis, and this can be the reason of poor quality of life for people who have this chronic disease. So now, going back to what I was doing, I needed to develop in the beginning some sort of review paper about the potential mechanisms of action, studying how dextran is taken up by macrophages and why delivery of the drug would be beneficial, and how this drug can prevent fibrosis. And it was very interesting because today, in my current state of understanding how science is done, I would look at this task very skeptically because there was like clearly lack of data, and I was left with only like speculating about the potential mechanisms. And of course, it's okay to build a hypothesis and speculate, but if it's too much of speculation, we're moving too far from something that potentially is true. So given that, I started to figure how to write a good review paper. And I started to read about like how to, I started to study how to write a good scientific publication. I took a Coursera course, Writing in the Sciences, actually developed in Stanford, and which is a very good course. So if you want to learn how to write better any of scientific texts, publications, I highly recommend. So anyways, I started to figure out how to write a good paper, and simultaneously I started to figure out how to do good science, which led me to understanding that in this institute the science is not that, like a high level, and publications were, there was lack of publications in international journals, and especially as a journals with high impact factors. And while there is widespread idea that like scientists who do publish with journals with good impact factors tend to say that it's unimportant, that it's like just a number that doesn't mean that much, and we should not hire or promote people based on number of citations, impact factors, blah blah blah. So all this passion to those metrics when we're evaluating someone's impact in science is like a little bit overrated. However, for developing countries and for the places where science is not that like high level, let's say no one in this organization have ever published in Nature. No one in this organization has ever published in journals with impact factor more than 10. So this is some sort of, not even a correlation, this is kind of association. And if your mentor, if your scientific advisor never published in a very high quality recognized journal, chances that they can teach you to do that are extremely low, disappearing chances. And this means you either learn it by yourself, if of course if you have such an idea in mind that you won't develop yourself as a scientist. So you either learn it on your own, or you switch organizations. And at this point, I really liked this challenging task, and I was like, okay, I will learn it by my own. And now looking back, I think that was a stupid decision. It wasn't the smartest one, and I made a bunch of strategically suboptimal decisions. Let's call them suboptimal. But this made me to rely on myself, on learning, and I took in parallel the third job. So I had this job in the institute, I had the job in marketing, and I took another job in scientific translations. So I was doing translations from Russian into English, and this publishing house, they were providing me with the feedback, like how their higher level translator reviewed my work and improved it, so I could learn better like what in my translation has to be improved. So I was doing it for several years. It was very, very low paid job, but it was all for learning. So I had this task of figuring out why dextran had this anti-fibrotic effect, and probably anti-fibrotic effect independently of mycobacteria, or maybe in a connection with mycobacteria. So I had to find the potential mechanisms. I started to figure what if dextran is picked up by the macrophages, is it mediated by some certain receptors? So I figured the number of receptors in our body that can bind the dextran, and started to learn what are the roles of these receptors, just generally, and in connection to infection. And I figured that the same receptors macrophages can use to pick up mycobacteria, and those receptors not only help to pick them up, those bacteria, but also do some sort of immune modulation, and they shift the response of macrophages to the bacteria from more pro-inflammatory towards more anti-inflammatory. And we know that fibrosis is a program that is associated with anti-inflammatory responses in macrophages, at least in macrophages. So they are pro-inflammatory M1 macrophages and anti-inflammatory M2 macrophages. And these, let's say, like general subtypes of macrophages, of course, now we know that actual science is different, but this is like traditional view. But these macrophages support generation of T cells, also with specific functions, like M1 macrophages are supporting Th1 cells, T helper type 1, and these cells support responses against intracellular pathogens. And this is any viruses and some types of bacteria. And mycobacteria are intracellular pathogens. So my idea was, okay, if dextrans occupy those receptors, and now bacteria cannot interact with those receptors, cannot induce pro-inflammatory response, cannot induce anti-inflammatory response. Now they tend to induce more pro-inflammatory response, and in parallel, they less drive the overall response towards fibrosis and rather drive towards inflammation. So that was my hypothesis, and I was trying to push it up and get resources to test it. So in parallel, I started to figure out that while we were hired, the lab capabilities were not built. They were built in parallel, and they were built super slowly, and all the reagent ordering system in Russia was broken, and it's still very much broken. Like, you need to order the antibodies, you receive them in 3 months. And after a while, I tried to push it, I tried to push at least publication of the review, and in the end of the day, I felt so much of misalignment that I was about to leave this place, and but I needed some alternative way. So I found in Novosibirsk there was a tuberculosis research institute. So I connected to people there, and I told them, look, I have a hypothesis how inexpensive, well-tolerated drug dextran can potentially change immune response in tuberculosis. And here is a very mechanistic hypothesis, and I know how to test it relatively cheaply using in vitro experiments, and I suggested them to try it out and build a small project. So they hired me. So I left my previous organization after spending 3 years there. I really tried to make it work over there, but it didn't. So I had to switch organization, and it always is bad for the PhD project. It cannot be done on time, and like, I needed to start everything from scratch. And for people who get into PhD projects, PhD programs in US or in Europe, they would be like, what the hell is going on? Like, how is this even possible? Person in 3 years ran, I don't know, a dozen experiments. They did not set up a lab for 1.5 years, and when they did, they started to work super slowly. In the end, they decided not to go with the project and kind of delay and eventually close it. Like, this is crazy, and this would be seen as lack of responsibility of the leadership, and at some point I think it was. But no big deal. I switched the institution, and at Tuberculosis Research Institute, I had amazing support for this project. Simultaneously, there was no good funding for it, and building the lab took a lot of time, so I needed to collaborate with different other labs. So I found different institutes to collaborate within Russia relatively quickly, within Novosibirsk, my home city, and relatively quickly I figured that I also would very much benefit from collaborating with international researchers, and I joined international societies where I wanted to submit at least my thesis, my abstracts. So one of them was EACI, European Academy for allergy and clinical immunology, or European. Yeah, anyways, EACI. So I found a mentorship programs there, and I met with a mentor, Stefan Martin, who really helped me to make this review look better. I had a chance to visit him. I had a chance to see like mouse preparation, how to find the lymph nodes. It was super exciting visit to Germany, and it was in parallel with going to the conference. So this was exciting, and then later I joined ASM, American Society for Microbiology, and I eventually I submitted my abstract to ASM conference, so I was presenting there. Now, looking back, I see that the quality of science that I was presenting was absolutely uncomparable to what students, grad students were learning in good universities here in US or somewhere in Europe. And it is a lack of resources, and it's a lack of guidance. And I think that many scientists who would become good scientists, but they grow up in some distant places, and they cannot join strong laboratories, and they cannot connect to strong scientific mentors. They just end up like not being able to build a good scientific career and to build a good scientific background. So, but anyways, I managed to come to US for half a year of joint research with Pooja Jain's lab at Drexel, and this really helped me to push this research forward. I eventually published the review paper, and I eventually published the short research paper, because at this point I already figured that the same receptors are used by HIV to enter the cells. So I had a parallel idea that what if dextran, extremely cheap and well-tolerated, approved drug in humans, can block HIV entry? Can we somehow use this idea of like blocking entry for the virus to achieve anything useful in clinic? And the results were showing to me that most probably not, because we were preventing viral entry, let's say like 4 times, and okay, 4 times less viral particles enter some specific cell type through some specific receptors. What would it give? Literally probably nothing, because viruses, they multiply very quickly, and however, I was ready to I was able to publish this, and sometimes negative results is also some sort of results, and it was not absolutely negative. Hypothesis that we can block the entry, it worked. Yes, we can. It's just not very efficient blockage. So after that, while I was next to Philadelphia working at Drexel Lab, I connected to some other professor who invited me for the postdoc. So right after I finished my PhD, I went for the postdoc in Philadelphia. And there I have a little bit of turbulence. I joined the wrong lab, or like we were misaligned, and I had to switch the labs. And after I switched to Immunology of Cancer. And then starting to work in Immunology of Cancer was extremely interesting, because chronic infection has a lot in common with cancer, and it is constant immunosuppression that allows some infectious agents to stay within the body without being destroyed. And similar story happens to the tumor cells. So they find some ways to suppress immune responses against cancer. And for the case of chronic infection, those infections, they have advantage of thousands and millions of years of evolution to adjust and build the ways to suppress or prevent immune responses. However, we as mammals, we developed evolutionary different ways to not let them avoid immune suppression. So they have a lot of mechanisms and we have a lot of mechanisms to eventually discriminate and destroy them. In case of cancer, it's slightly different. In case of cancer, cancer cells start to develop some sort of mutations, some sort of aberrations, and normally early on there would be no immune response to the cancer cells. But once which start to grow and start to adapt, they start to adapt and develop those mechanisms of immune evasion. And our body, in the beginning, there is probably less means to recognize that newly originated tumor cells, but after some time, those tumor cells are less protected compared to like infectious diseases which developed like many mechanisms through evolution. They're less protected from immune system. But after some time, the cancer that grows into serious disease, it finds way to avoid immune response. This is why it grows. And immune oncology, it was 2017, immune oncology was already very hot at this point, and I understood that if I want to move towards industry, it's a better place to be than immunology of infectious disease. And at this point, no one knew that there will be COVID 2019. It was only 2017. So, all right, what's next? And immunology of cancer. When I was learning immunology of tuberculosis, I knew how important Th1 response is. Response of the T cells against intracellular pathogens. And the same response is important against viruses. But when I moved into cancer immunology, I saw that we're not only interested into the specific T helper cell responses, which are more properly called CD4 cells, just because it's easier to study them and know that this marker CD4 is we describing the CD4 cell population instead of all the term T helper cells. So not only they are important, but also CD8 cells are important, which are T killer cells, as in traditional terminology. And like there are ways to induce immune response against tumor by using different means of vaccination against cancer. And I really love these concepts. I was reading a lot, I was running a lot of experiments. And this is where I saw how proper, like quick, streamlined series of experiments are organized. I was running multiple projects in parallel. I had access to a lot of reagents. Some antibodies were coming the next day. It's amazing. It's amazing. It was an amazing opportunity to work very intensively, experimentally. I had lack of training in running so many experiments, which were many of those models were new to me. But I was learning rather quickly. I was generating some results. I would say in this postdoc, I was kind of unlucky. We tried a lot of things, and very little number of them worked. We were trying to stretch to work on very like risky ideas. And during my postdoc, I was figuring out what type of career I want to build, because staying in academia, with my lack of good background in generating great science, because in Russia I had no opportunity to publish continuously in good journals, and I was self-leading my PhD. I was building the lab on my own. I was helping to create the mouse facility on my own. It was like a very unique case, but I was not prepared for academic career in US, which is very competitive even for people who are coming from this same system. A number of professorship positions is extremely low, and I would say the balance between what the young professor is giving to university and giving to science is kind of disproportional to what these people get. Of course, if they value this like scientific freedom, which is also not a complete freedom, it might be great for them, but I understood it's not for me. And I was looking at different potential career directions. I was looking very widely across the board. Like, do I want to do consulting? Do I want to do IP law? Do I want to become an application scientist or research scientist? Do I want to go into maybe something even close to technical sales, or do I want to I was looking at many different options. Do I want to go into tech transfer office? And I was thinking, can I at some point start my own company? And what line of career will prepare me for this? And I had informational interviews with people from many industries. And I ended up thinking that becoming an industry scientist is the best way for me to go. And what helped me to even start this journey in understanding where I want to go in my career, it was Cheeky Scientist Association. And when I was doing my PhD, I was watching those webinars ran by Isaiah, and at those old times there was less selling in those webinars, and there was more of great career advice. And I think that now it's a little bit too much selling, but don't get me wrong, I had something to compare it to, and early webinars were like, I felt that this guy wants to help PhDs to get hired into industry, because finding the good academic position was extremely tough, and those positions were not quite rewarding, especially postdoc positions for Americans. Like, not too many Americans go and do the postdoc. So many international postdocs are working in universities because those positions are not considered lucrative. They consider like being a cheap labor, even if it's sophisticated labor. So I started to figure out where I can find the right match into industry, and in what type of industry I can keep building up the career to maybe someday start my own company. And I think that in the next video I will tell a little bit about my first industry job and how it led me to creation of a company. And this will be third step, like wrap-up step, why I started 100x Bio, how this idea even came to my mind. All right. For everyone who is subscribed, see you again, and thank you for watching, everyone. Have a good one. Bye-bye.